Hello, class. Hi. How are you guys doing? My name is Toluleke Famuyiwa. I'm the teaching assistant for this course, Cell Biology, and I'll be taking today's class. Today's class is going to be on translation. That is the part that follows after transcription. Translation occurs in the cytosol. It involves the messenger RNA base pairs that have been translated into amino acid sequences. It involves the nucleotides in such a way that you're going to have them coming in groups of 3. When they come in groups of 3, that's what you call the codon. Evolution picked codon in such a way that we have 64 of these varieties. And in order for you to have this calculation, you're going to have to multiply 4 different bases with 3 bases per code. So in other words, if you do 4 raised to the power 3, in total you're going to have 64 codons. The relationship between this codon and the specific translation that you have is what you call the genetic code. And this is the example of the genetic code. So at the beginning you have leucine, alanine, all these amino acids in between. You have the start signal, and at the end you're going to have the stop signal. This is the example of stop signal. Transfer RNA molecule. What is this about? Why do we have to talk about transfer RNA molecule? The transfer RNA molecule forms the link between the codon, the amino acid coming in groups of 3, and the amino acid molecule. Transfer RNA consists of 80 nucleotides inside in such a way that the anticodon segment of the transfer RNA will bind to the codon on the messenger RNA, such that there is a lock and key device going on there. Specificity, just like we've been discussing. The transfer RNA molecule is synthesized by polymerase 3 as a large transcript initially. Then you're going to have the trimming of the ends and also the trimming of the middle part, which we know as the introns. Some bases are chemically modified to yield unique structures. And this modification defines the final conformation of the particular protein that you're going to find. So the transfer RNA looks like this. For example, you have the attached amino acid, in this case the phenylalanine. It has the 3' end, the 5' end. Also the T loop on this side, the D loop on this side. Then we talk about the anticodon loop. This is very important because we said that this anticodon loop is the one that is going to bind with the codon on the messenger RNA. And this is an example of how this looks like. You have anticodon on the transfer RNA, which is going to bind with the codon on the messenger RNA. In order for this process to continue, we need the help of amino acid synthesis in such a way that the amino acid tryptophan will be linked to the transfer RNA that is specific to tryptophan with this transfer RNA synthesis in the cloud. When this process continues, what are you going to have? You're going to have the use of energy to form a bond. This bond is going to be formed between the tryptophan molecule and the transfer RNAs for tryptophan. And we know that this I energy bond will be used later to form the peptide bond by the help of the large subunit, which we're still going to be discussing after now. Hydrolytic editing. Why is this important? During the formation of the protein, at the last stage of translation, there is a possibility to have an error in such a way that there's incorrect amino acid being used. When this happens, that means the synthesis is not done. There is need to do editing in such a way that this incorrect amino acids will need to be transferred to the editing site. When it gets to the editing site, then the incorrect amino acid will be removed and a proper amino acid will be attached. And that's what you call synthesizing and eventually editing. Ribosome. We talked about ribosome earlier. We said ribosome is a large ribonucleoprotein complex composed of two main subunits. The first is the ribosomal RNA, and also you have another component that is the over 50 proteins. So the coming together of this is what you call the ribonucleoprotein. Each subunit is assembled in the nucleolus as seen in the last class, where the professor was showing the structure of the nucleolus. He was talking about synthesis versus assembly, if you guys remember. In eukaryote, ribosomal RNA component is transcribed as pre-RNA. That means you don't go directly. You go to pre-RNA first before you go to RNA in eukaryote. Two main subunits exported into the cytosol, where you have millions of ribosomes per eukaryotic cell. This is how ribosome organization looks like. You have the small subunit that contains messenger RNA binding site, and also contains the site for tRNA, because both are very important in this translation stage. The large subunit is going to come in, like I said earlier, it's going to catalyze the formation of peptide bond. We know peptide bond is very important in the organization of this structure of the proteins that we're talking about, because we talk about hydrogen bond and peptide bond. Eukaryotic initiation, how does this look like? The initiator aminoacyl tRNA loads onto small ribosomal subunits, also known as SRS. So whenever you see SRS, we are referring to small ribosomal subunit. Then eventually you're going to have addition of eukaryotic initiation factors that will bind to the SRS. The small ribosomal subunit then binds to the 5' end of the messenger RNA. But the one thing you need to note here, very important, because with my experience I see that this could be tested. In eukaryote, you have this. But in bacteria initiation, there is no 5' end interaction. The way I study is to have the tabular form such that I'm comparing already the differences between what is going on in the eukaryotic state and the bacterial state, because we're still going to see that as we go on. Eventually, in the eukaryote, you're going to have the recognition of the 5' cap. The small ribosomal subunit then scans for messenger RNA using the codon AUG. Okay. The movement is powered by ATP hydrolysis, because we know that for such movement to occur, energy is needed. Energy is provided in the form of ATP. Eukaryotic start looks like this when the start codon is encountered. There's going to be dissociation of the eukaryotic initiator factors, the large ribosomal subunits will bind in an attempt to facilitate the production of the peptide bond. The first AUG codon encountered is usually not the start. Just like the first signal that you get in life is not usually the best signal. It could be the best signal, but it doesn't have to be. And in this case, when the first two signal is not going to initiate the start, we call this phenomenon leaky scanning. The whole process allows synthesis of different proteins that can be identical in function. But what happens is that they have different destinations based on their tip. For example, the same protein is produced based on the information on the tip. One protein can go to the lung, to the lungs, another to the kidney, another to the brain. So that's making you know that whatever they have on the tip will determine their destination in the body. The eukaryotic start looks like this. We have the eukaryotic initiator factor with high energy already. The initiator tRNA is in. The small ribosomal subunit is now binding to the initiator tRNA. Then there's addition of initiator factors going on here. Then we said the SRS is going to look for the codon AUG on the mRNA. When this is located, that is when the initiation process starts. The initiator tRNA moves along the RNA searching for the first AUG, and we said this process needs energy. That's why ATP is coming in, and you have the hydrolysis of ATP going on there. Then eventually we said the large ribosomal subunit is going to come in. Amino acyl tRNA binds because their help is needed in this process. Eventually you're going to have the formation of the first peptide bond, and that is the end of the eukaryotic start. Moving to the bacterial start, how does this differ from the eukaryotic start? For the bacterial start, we said the messenger RNA have no 5' cap. Again, this is the second time I'm saying that. There's a special consensus sequence that signals the induce of SRS to begin the scanning for the codon AUG. So the signal in bacterial start is different from the signal in eukaryotic start. Many consensus sequences and associated AUGs per bacterial messenger RNA. Bacterial messenger RNA are polycistronic. What do we mean by this? Poly means many. The question is, many of what? Several different proteins are synthesized from different regions of the same messenger RNA. The polycistronic bacterial messenger RNA looks like this. The same messenger RNA, the 5' end, the 3' end, you have the ribosome binding site. Then you have the synthesis of different types of proteins. In this case, 3 different types of proteins. The first one is the protein alpha, protein beta, and protein gamma. Then we move to the elongation step. This is the growing polypeptide chain, the incoming amino acyl transfer RNA. This is the P side and the A side, the 5' end and the 3' end. The first thing that happens is this bond is going to be broken, such that this 3N that is attached to this is not going to be there anymore. The energy from the broken bond is used to form a new bond, such that we're moving from here, the connection between this 3 and this end, to the connection between 3 and 4. The conformational changes shift the large subunit to the right. We can see that here the large subunit looks like this. Eventually, it's not directly on top of the small ribosomal subunit. It's moved a little bit to the right. While this is going on, the small subunit, the messenger RNA and the transfer RNA do not move. They stay at the same location. Elongation step 3, the small subunit and the messenger RNA move back underneath the large subunit, while the transfer RNA do not move. This is difficult to see based on what you're looking at, but know there's a movement going on there. Then what happened is the unloaded tRNA in the E side is displaced, because when something is loaded, it's firmly attached. It's not really loaded anymore. It's going to be just hanging there. While it's hanging there, then it could be easily displaced. So that's what is happening there. The elongation factors. So this diagram is telling you what the function of the elongation factor is in this whole process. The elongation factors come in with the high energy molecule, GTP. Before you know it, there's incorrect base pair tRNA that are being dissociated, because we said that if the tRNA is not pairing well, there's opportunity to replace that tRNA with the correct one. Then eventually energy is needed for this process, and when the GTP molecule is broken down, you're going to have GDP eventually at this end. The stop codons, for example, will look like this. The UAA, the UAG, and the UGA stop codon. We said, for the most part, AUG is the start codon. The stop codons bind release factors. And what happened is that these release factors function almost like the amino acyl transfer RNA. Because of this similarity in structure, we said there is molecular mimicry. That means one structure actually pretending to be doing something that is similar to a second structure. That's what we call it, molecular mimicry. If you've done biology in other sections, you've had the word mimicry before, but this is a little bit modification because it's happening at the cellular level. We are calling it molecular mimicry. The release factors had water molecule instead of amino acids. So the amino acid was being added earlier, but now you are pouring water to the whole structure. Eventually, when water is added to chemical reaction, we call it hydrolytic reaction. You're using water molecule to break a bond. As a result of that, water addition frees the last added amino acid. The amino acid was expecting an amino acid to be added. Amino acid, another amino acid, another amino acid, another amino acid. But instead, water was added, and as a result, the last amino acid will follow. Translation termination. How does this occur? We mentioned just now the binding of release factors to the A site. instead of addition of amino acids, there's addition of water molecule, eventually leading to the falling off of the polypeptide that is produced. As a result of this, the small ribosomal subunit is falling off, the large ribosomal subunit is falling off, then the release factors is also being thrown away, and this can be reused again. Polyribosomes, what do we mean by this? Poly means many of ribosomes. In other words, we are talking about multi-ribosomes that operate in a tandem function, like one after the other. That's what tandem means, one after the other, like that, on the same messenger RNA. It increases the rate of protein synthesis in both the prokaryote and the eukaryote. The bacterial polyribosomes, another major difference you need to note now is that when the transcription process is going on in bacteria, the transcription process is going on, is starting from this end, going to this end. In bacteria, as you're moving, you can start translation. It's like somebody that can do two things at the same time, right? Writing the notes and also preparing for another class. Some people can do that, some people cannot do that. In the case of bacteria, you find out that transcription is going on, translation can go on at the same time. But in eukaryote, that is not the case. Transcription must be completed in eukaryote before translation starts. The eukaryotic polysome looks like this. You have the messenger RNA with the start codon, AUG. Everything keeps going on till you have the stop codon at the end, when you're going to have the termination. Efficiency of translation. We said in life is about trade-off. You gain something, you lose something. You're not married before you become married. You have companionship going on, but you've lost your freedom to do certain things. That's what life is. You can't fight it. In the same way, in the cellular level, it's the same. Evolution is in between picking accuracy and speed. Like, you either pick the accuracy or the speed. You cannot pick both at the same time. You cannot be tall and be short at the same time. You cannot be fat and be thin at the same time. And this is not without exception in protein synthesis. Evolution decided to pick accuracy over speed during protein synthesis. This involves the large amount of energy used to ensure accuracy. In order to achieve this accuracy, we need at least 4 molecules of ATP per peptide bond. And for our record, this is the highest number of ATP required per peptide bond. This is making you know the importance of translation to our body. It's very important, because if there's no protein, you can't sit down like this. You'll probably be on the ground and doing whatever that is so funny that you feel like doing. We can only sit down like this because we have proteins in our body that are functioning properly. Then we have to talk about protein folding after the protein is produced. All secondary and most tertiary folding occurs as synthesis proceeds. Like the proteins are produced, the proteins are trying to fold, produce, fold, produce, fold. That's what is going on there. But we need the help of molecular chaperones just to mediate and speed up this tertiary folding, which may also involve correcting some defect as a result of the folding. Molecular chaperones that are important in this case, we have two families of these chaperones. The first one is the heat shock protein 60, and the second one is the heat shock protein 70. The heat shock protein 70 binds to the hydrophobic regions and uses ATP to assume different conformation in order to say, well, this is not so good. I want to reverse something back. It needs energy. And that's why ATP comes in at that point. That forces incorrectly folded proteins to unfold and allows them to attempt refolding. So you have a folding that is really wrong, just like this, right? The first energy is to make sure that it is unfolded, and the second energy is to see whether it's going to be folded the proper way. Just like you have friends that will get out of their bed, will not make their bed, they just squeeze the bed sheet and throw it on the ground. And you have friends that when they got off their bed in the morning, they will fold their sheet very well and put it back. The heat shock protein 60 forms a barrel-like structure. We think of a small container that can house the protein structure. It provides misfolded proteins an isolated environment to attempt refolding. The heat shock protein 60 and the heat shock protein 70, this is how they look like. So the red part is the heat shock protein 70. It comes in with the help of ATP energy. It helps this structure to unfold first. Then with help of energy again, it tries to fold again, which sometimes it can be correctly folded or even incorrectly folded again. In this case, you have the heat shock protein 60 coming in form of a barrel structure. And it's going to be hydrophobic binding at the binding site. So picture we are in a house, somebody cooked the rice and the rice is not cooked very well. Another person have to come again to cook the rice to make sure it's cooked very well. So the second cooking is what is going on here, whereby you're going to have the structure coming in again, the plate is covered, energy is going to be needed in any form. In the case of cooking, it's in form of heat. Then energy needed again to attempt refolding. And sometimes you're going to have the folding being corrected as a result of the energy that is applied. Quality control. Proteins which cannot be refolded by the chaperones, heat shock protein 60 and heat shock protein 70, something needs to be done about them. So what happens is these are digested by proteasomes. These are special cases of proteins that degrade large peptides into smaller peptides. The very big ones that are just useless, you try to unfold them, you cannot unfold them. So the next thing is to break them into little chunks. The short peptides further degrade in the cytosol by proteases, and misfolded proteins are targeted by ubiquitin attachment in such a way that this ubiquitin are proteins that they have the ability to help degradation of proteins that are defected in such a way. In this case, they are defected in form of their shape, because we said that in order for protein to be functional, their shape is very important because that's where everything comes in. The electrochemical environment, conformational change, the shape, and they're going to have specificity to where they are going to. That's what is going on there. Multi-ubiquitin chain looks like this. The ubiquitin activating enzyme is the E1. The ubiquitin in the red color comes in. Energy is needed. There's going to be binding of the ubiquitin to the ubiquitin ligase. You have the structure coming together here where you have the E1, the E2, and the E3. The E1 comes out. Now, as a result of that, you have ubiquitin ligase primed with ubiquitin. It's a structure that is going to allow us to be able to see the presence of these proteins that needs to be degraded. And this protein is going to have the signal on it. And this is the signal that is indicated on this top like this. As a result of this, this ubiquitin ligase complex will come in to join with the targeted protein that has the signal on it. As a result, the ubiquitin will jump off this complex and it's going to come on the protein. Because that's what we want. We want the ubiquitin to go on the protein. And when this happens, you have multiple ubiquitin on the targeted protein. Eventually, this is going to degrade the protein that we don't want. We don't want the protein anyway. So it's just going to be like little pieces there. This is the end of the lecture. On Tuesday, there's going to be the second exam. It's going to be February 26th. The exam is going to start at 9:30 a.m. You need to come with your small green Scantron. The professor is going to pose some sample questions later today or tomorrow. I'm not sure. But we're going to have review session today from 11 a.m. to 12 noon.
Can you start the review session now?
That's kind of tricky because some people left.
They left. So?
They left because they probably want to come to the review session. You guys are so smart. You can put me in trouble because of that. I'm a student also, so don't try that with me. I know students. They say, oh, well, I just have to attend to a family emergency. You posted on the canvas that the review session is going to be this time, and you're going to change the time. You should let everybody know. I'm not going to do that. I think we should do it over there because there might be a class here.
Well, this class ends at 10:50.
Yeah.
If you start now, you still have an hour to do the review. Just a little bit.
So you want me to do a review session twice? Why should I do it twice? No.
Well, because here's the thing. If you go to the place at 11 and no one shows up, then you can leave early. Because you already did the review.
I don't do things like that. I'm sorry. We have to be considerate. I don't think it's fair.